Okay. That's right. Okay, so DNA replication, you guys know based off of what's on your paper there that this actually says replication and that it just looks like it says replication because there's a because there is a hole there because I just copied what y'all have in front of you there. Okay, so we're going to talk through kind of the high points of what happens during DNA replication. So what is DNA replication? What are we doing here? Making more DNA. Is it different from the original strand or the same?
Same.
It's going to be the same. We're going to have two identical strands at the end that are both identical to the original strand. The two strands of DNA that we get at the end are each half old DNA, half new DNA. Okay? So the strands that we get at the end are each half old DNA, half new DNA. Everybody's got that part? Okay, so here's our DNA here. All right, and just to, for a frame of reference for us, I'm going to use different colors so that we can remember what's the old DNA and what's the new DNA. Okay, so here's our original DNA. I'm making it purple because I'm pretty sure that's what I did yesterday. So here's our original DNA. We want to make an exact copy of it. So what's going to happen is helicase unwinds it. Okay, so it looks kind of like a ladder. Still old DNA. We haven't brought any new DNA in. It's going to unzip. Okay, which exposes those nucleotides. And y'all see here that I kind of wrote out some nitrogenous base pairings just to give us a frame of reference. And this isn't a code for anything in particular. I just came up with a repetitive code. So this was kind of where we left off yesterday. So helicase unwinds, unzips the DNA. We talked about how the DNA is anti-parallel. So that like we have the 5' end of one side of the DNA and the 3' end is at the other end, and then the complementary strand is going in the opposite direction. And remember that it has to do with the direction that the sugars are pointing, right? Okay, so this end is called the 5' end because the phosphate is attached to the 5th carbon here, and this end is called the 3' end because the phosphate on this end is attached to the 3rd carbon of the sugar. So remember that it's just based off of which direction the sugars are facing, and that the two strands are just opposite of each other. All right, so that's where we're at now. So we unwound and unzipped the DNA, and we understand that the strands are anti-parallel. So now we're going to line up new nucleotides on both of these sides of the DNA in order to make two new strands. So what did we call the strand that moves really fast through replication? Okay, the leading strand. Then we've got the lagging strand, which kind of lags behind just because it's more difficult for the lagging strand to go through the process. It just takes longer. There's more steps. Okay, so what I did was I made step 3 is what's happening to the leading strand, and then step 4 is what's happening to the lagging strand. And this is actually happening simultaneously. I just separated it so that it's easier to visualize. Okay, so for the leading strand, an enzyme called DNA polymerase 3 is bringing new nucleotides over to the leading strand. Okay, so you can see here there are a bunch of free nucleotides. They're just kind of hanging out. Okay, and DNA polymerase basically picks them up and brings them over to match up with complementary DNA. Okay, so on the leading side, we have a T there at the bottom. Okay, so DNA polymerase 3 is going to bring an adenine nucleotide over to match up with it. Okay, why are we starting at the end away from the replication fork? True, yes, but why? Look at this. Let me read what I wrote here to the right. New nucleotides are always laid down from a 5' end to the 3' end toward the replication fork. Okay, so that's really important. So we start over at the very end here because we're laying down from the 5' end and we're working our way towards the 3' end of the new DNA strand side, right? Okay, so once we're finished with it, this end is going to be 5' because the sugars are pointing in this direction, and this end is going to be 3'. So that means we have to start on the what's going to be the 5' end, and work our way towards the 3' end. and we always have to be going towards the replication fork as we're laying down the nucleotides. Okay, so those are the two main points. One is that the new nucleotides have to be laid down from the 5' direction to the 3' direction, and two, that has to happen moving from the outside the replication fork towards the replication fork. And then, why do y'all think that is? What's going to happen at the replication fork as soon as we've filled in what's already unzipped? It's going to unzip a little more, and then we can work our way towards the replication fork again, right? Okay, so that's why we're always moving towards the replication fork. All right, so let me kind of fill in all my old DNA so that I can go back and get a different color for new DNA. Okay, so new DNA, I made it red yesterday, right guys? Okay, all right. So all the new nucleotides are being brought over, they're being matched up, okay? And once we get plenty of all the nucleotides lined up, then they're going to be bonded together by an enzyme called ligase. So I need to bring like a cytosine over here, a thymine, a guanine, okay? And once I get all those lined up, then ligase is going to come in and it's going to bond those nucleotides together to make them a chain like what we see down here at the bottom left-hand corner, okay? So what we did here was all these new nucleotides were lined up in a row, and then ligase bonded them together so that they're a complete strand. Okay, so that's what's happening at the leading strand, working from the 5' end to the 3' end of the new DNA. All right, so while that's happening, the lagging strand is also like adding nucleotides, but it's happening in a different way. It's a lot more complicated just because of the direction that the sugars are in. So let me read through these steps and then we'll come back to the picture. In order for the lagging strand to lay down in a 5 to 3 direction, moving toward the replication fork, the process goes like this. First off, an enzyme called primase lays down a primer, okay? And it's gonna lay it down just a few nucleotides in, okay? And then DNA polymerase 3 is going to lay down what are called Okazaki fragments, which are segments of new nucleotides that match up, okay? And then DNA polymerase 1 comes and picks up that primer that was laid down earlier and takes it away, and ligase binds the Okazaki fragments together. Okay, so let me kind of show you, everybody make sure you're looking up at my screen for just a second. Okay, so this is the leading strand, it's doing its thing. All right, so now we're at the lagging strand. So we still have to move from a 5 to 3 direction towards the replication fork. So what we're gonna do is, so that we can do that, is that we're gonna do it in sections. Okay, so what's going to happen is that primase, make sure you're looking at my screen, primase is an enzyme that's going to drop off a primer, and it's going to like leave a little space here at the end, and then primase is going to lay a primer down like right here. And basically that's just giving the new nucleotide something to bond to. So then DNA polymerase 3 is going to bring in this set of nucleotides that match up called an Okazaki fragment. It's going to lay those down in a 5 to 3 direction. It starts here and goes dun, 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 dun. Okay? All right, so now we have a section. Now, that's going to happen again. We put another primer here, and then DNA polymerase 3 comes and lays down another Okazaki fragment. Okay, so now we have 2 Okazaki fragments next to each other with a primer in between. Okay? So we have all the DNA we need, but now we have a primer in between, and so that's kind of a problem. So DNA polymerase 1 is going to go pick up the primer, and ligase comes and bonds them together. Okay? All right, so I'm going to show that video again because I think that it will help for us to see the video again. Let me stop the recording.